Hello, everyone. I'm very pleased to introduce the paper entitled Comprehensive Lung Microbial Gene and Genome Catalogs Assist the Mechanism Survey of Mesomycoplasma hyopneumoniae Strains Causing Pig Lung Lesions. It has been published in iMeta recently. Respiratory diseases which influence pig growth and feed efficiency and result in significant economic losses are considered one of the most important health problems in pig production. In the past few years, growing evidence has suggested that the lung microbiome has a significant impact on respiratory diseases. It is thus critical to reveal the community structure of the lung microbiome to explain its roles in maintaining lung health and in related diseases. In this study, we performed a deeply metagenomic sequencing analysis on 744 microbial samples collected from the lower respiratory tract of 5 pig populations. The main highlights are as follows. First, we constructed a comprehensive microbial gene catalog, which contained 10,031,593 non-redundant genes and obtained 356 non-redundant MAGs. Second, we observed the significant associations of Mesomycoplasma hyopneumoniae strain or adhesion-related virulence factors with lung lesions and confirmed this with in vitro infection experiments. Third, we suggested that the adhesion of Mesomycoplasma hyopneumoniae activated several pathways of bronchial epithelial cells, such as NF-kappa-B, MAPK, cell apoptosis, and response to reactive oxygen species, and resulted in cilium loss, epithelial cell-cell barrier disruption, and lung lesions. And now, I am introducing the main findings of this paper. First, we constructed a comprehensive microbial gene catalog of pig lower respiratory tract microbiome, which contained 10,031,593 genes. Based on this catalog, the taxonomic composition was characterized, including bacteria, archaea, fungi, and virus, as well as potential function. Second, we recovered 1965 MAGs using the metagenomic binning approach. These MAGs were further clustered into 356 non-redundant MAGs and 256 SGBs. Among these 256 SGBs, 41.8% were defined as unknown SGBs. 613 F7 pigs from the mosaic population were divided into 4 groups of healthy lung, slight lung lesion, moderate lung lesion, and severe lung lesion according to their lung lesion scores. We then investigated the relationship of the lung microbiome with lung lesions using the constructed gene and genome catalogs, and observed the distinct significance of the correlations of different Mesomycoplasma hyopneumoniae strains with lung lesions. We used 291 genomes of Mesomycoplasma hyopneumoniae for pangenome analysis. 2 strains were selected for RNA sequencing analysis, compared with the 168L strain with weak pathogenicity. MAG47-like strain with high pathogenicity had no significant difference in the distribution of virulence factors, but the expression levels of virulence factor genes encoding adhesins and adhesion-related proteins, such as P97, were significantly higher. In vitro infection experiments show that MAG47-like strain caused a significantly higher number of damaged cilia and more severe damage to the epithelial barrier. Compared with 168L infected bronchial epithelial cells, MAG47-like infected cells had higher expression levels of genes FOS, SOCS3, MAP3K14, CD74, GAD45A and IRF1, suggesting that MAG47-like infection led to stronger immune and inflammatory responses, oxidative stress and apoptosis, which led to more severe lung lesions. In summary, we constructed a gene catalog that contained 10,031,593 microbial genes and recovered 356 MAGs. Based on these datasets, we found the heterogeneity in the microbial composition of the swine lower respiratory tract microbiome among individuals. We analyzed the pangenomes and investigated the distribution of virulence factors in Mesomycoplasma hyopneumoniae strains. We confirmed the effect of Mesomycoplasma hyopneumoniae adhesion on the lung lesions, and elucidated the possible mechanism involved.